This is our Science 9 reproduction project. On this page you will find everything about asexual and sexual reproduction. You will also learn about how plants reproduce both asexually and sexually, how chickens mate, and how a chicken is born. On this page there are various terms you need to know, everything about biology, what you need to know about reproduction, and everything about cells and how they duplicate, such as mitosis, meiosis, DNA, cells, mutation, clone, and genetic variation. How chickens mate and what you need to hatch a chick with and without the shell. Once the egg is fertilized and has been laid, it will need to be put into an incubator before it reaches 7 days old. Incubators should stay at around 37.5 degrees Celsius, and the humidity should be at 60 to 65% and 70 to 75% at hatching time. This page provides what an egg would normally use its shell for, such as protection and nutrients. There are also notes from a study done on this, which are very helpful. Notes on how an incubator works. This is our lab write-up for hatching an egg in a shellless culture. For our introduction, after seeing that it was possible to hatch a chick in a shellless culture, we did investigation into how animals, especially chickens, reproduce, how our body cells reproduce, and other subjects related to the experiment. We came up with a suitable means to hatch the chicks, such as a semi-permeable membrane in which the yolk would rest in, and an incubator that would mimic the heat and protection the egg would normally get. After answering some questions, such as how it would obtain a fresh supply of oxygen, we were happy with our blueprints for the experiment and research, and we attempted to hatch the embryos. For our hypothesis, we believe that we can hatch the chickens in a shellless culture by creating our own culture for the embryo. Here is a list of supplies we had, and the procedure. Follow it carefully, and make sure to disinfect everything, and be careful with the embryos, so that they are not handled in a rough manner. Here are some possible errors. On day 4, the incubator alarm was triggered, signaling that the temperature had dropped overnight. One of the eggs touched the saran wrap while it was being prepped for incubation, which may have contaminated it with 1 or 2 other eggs. Also, the handling could have been a little more gentle. When an egg drops below its normal temperature, its hatchability decreases. The embryos were not fully developed. They gave little sign of hope due to the fact that they were late in developing and did not develop at all. One egg managed to develop a heart, which was seen to be beating, but soon died. We concluded that the experiment was a success, due to the fact that we were able to create a suitable culture for the eggs, even though they did not develop very much. Human error was a large factor. For example, breaking an egg and possible contamination with the eggs would immediately halt progress. It was a win and loss, and we hope that this website can provide good information for those who would like to do it too. This is a page where you can see chick embryo development, the videos we had from November to December on the eggs developing, and our references. Thank you for watching. This is Ben, Callum, Rajdeep, Kate, and Brad signing off.